The nephrons are the basic anatomical units of the nephrons. Please paste the picture into your notebook that I gave you out last class or download from Blackboard. And we have 1 million, around 1 million nephrons in each kidneys. They are the ones that they form continuously the urine, and they contain two parts, the glomerulus part and the renal tubules. The glomerulus part is a network of capillaries inside a colored brown, and the other part is the renal tubule system coming all around that eye, surrounded by blue. So they are the ones that they form the urine, basically. They are microscopic size, so you will not see it in the, with naked eye, but under the microscope you can. Now let's go with the vessels. This first one is the interlobar artery that is between the pyramids. Remember that when we cover that, they break up into two parts, the arcuate arteries, and vertically gives off the interlobular arteries, and tinier vessels come off like the afferent arterioles, that they start to form a capillary network as a glomerulus. Let's talk about this capillary network. Capillary network, and coming out is the afferent arteriole. Now, what is so important about glomerulus capillary network? This is only one. Arteries goes into the arteries, or arterioles connect to other arterioles. Now, the main point is this is the unit that filters the blood. Get rid of all of the materials what is not needed for the body. How to achieve the filtering of the blood? The afferent arterioles diameter is bigger than the afferent arterioles diameter. Also, the vessels are arterioles and they have high resistance. The arteries, you remember the muscle layer in the arterioles, and they make a high pressure system over there. These two, the one and the two, leads to a high hydrostatic pressure in the capillaries that pushes materials like solutes and smaller particles than proteins into the space that surrounds the glomerulus, into the renal tubules. And the renal tubules are the stuff that surrounds, more specifically, the tubule that surrounds the glomerulus. The tubule is, we call it the glomerulus capsule. This part, this blue part that I'm shading it up, this is the glomerulus capsule, or it's called the Bowman's capsule. We will use the Bowman's capsule more often, that name, honoring the person who just discovered that, or who just realized what it is. And get more closer view of the Bowman capsule on the side. It has a, so the outside of the Bowman capsule contains tightly fitted epithelial tissue, simple squamous, but the inside that is proximal to the capillaries are called the podocytes. Podocytes are loosely fitted cells with some tentacles that they attach to the capillaries, and they make this epithelium kind of a porous, so let the capillaries material get into the Bowman capsule, into that place to be able to form the urine. So after the Bowman capsule, we will have a very, very coiled tubule formation, and it's called the proximal convoluted tubule, or tube, or tubule. Tubule, it's more precise because it's kind of very tiny. We have another very coiled tubule. This will be, actually, no, it will go into a straight down and up tube, and this is loop of Henle tube, and terminates into a distant convoluted tubule or tube. This distal convoluted tubule will drain into this orange pipeline. This is called the collecting duct. This is what the urine will leave the nephron. This collective duct, remember that the pyramids have a striated appearance, and these are the ones that give the striated appearance for the pyramids, and they move into the minor calyces through the papillae of the pyramids. So the efferent arterioles will go around the tubules and turn into vanules, and they will be the interlobular vanules. They collected into interlobulars. The interlobulars move into the arcuate veins, and the arcuates will move into the interlobar veins. So let's talk about another capillary network, and the second one will be the peritubular capillaries. What is really the point that the capillaries that surrounds the proximal convoluted, distal convoluted, as well as loop of Henle? So this is a low pressure area because they are vanules, most of them. They are around the tubules, and they do absorption from the filtrate that got filtrated into the Bowman capsule for absorption of the solute and water from the tubules, because we are not getting rid of all of the material that is going to be filtered out into the Bowman capsule from the capillaries. So the histological stuff, the proximal and the distal convoluted tubules are made up of simple cuboidal cells, and the loop of Henle is made up of simple squamous cells, if we, by chance, would have a time to check it under the microscope. So far that's it, and we will discuss more in class.